Thank you for the invitation and nice introduction. This workshop is very special for us because it deals with sex difference in many different kind of neurological disorder. Today, I'm going to talk about the pathophysiological mechanism of sex difference in Alzheimer's disease. Firstly, I'm sorry to say that the slide book is quite different from what I'm going to show, because I realized that today's talk is 15 minutes, but my previous slide was about 1 hour amount. So last minute I changed the slide a lot, so my slide might be quite different from what you have right now. As you know very much, the Alzheimer's disease patients are mostly women. It's 2/3 of patients are women. Also, the higher risk of developing AD in women compared to men is about twice. Here is the some quantification, and as you can see here, this purple color is the woman percentage of the patient. So it's about twice, exactly twice more than men. Alzheimer's disease have various different reasons to cause those disease development, including obesity, diabetes, environmental factors, smoking, depression, sleep, genetics, hypertension, lipidemia, etc. In addition to these many different kind of reasons, the gender is most important factor to contribute to AD development. The Alzheimer's disease major pathophysiology is three things. One is extracellular deposited beta amyloid. Second thing is hyperphosphorylated tau, which made the intracellular neurofibrillary tangles. And recently, astrocyte and microglia also contribute to this Alzheimer's disease pathogenesis. Altogether, they started to kill the neuron to disconnect neuronal circuit, causing cognitive dysfunction in subject. Do women live longer than men? Yes, but clinical implication of sex difference in AD pathology is quite very significant, because as I told you, beta amyloid and tau are very important pathological things, but at the first at the first stage, disease stage, beta amyloid accumulation is very similar to female and men. However, as disease progress, female tau pathology become much faster than men. The woman is much faster than men. And finally, those clinical function, female has more and more severe symptoms rather than men. So in this case, show that women suffer the disease a lot compared to men. Also, women live longer than men, which means women has much higher the old age suffering time than men. In addition to beta amyloid accumulation and tau phosphorylation and aggregation, there are diverse pathological mechanisms of AD. For example, mental dysregulation, mitochondrial dysfunction, vascular dysfunction, and neuroinflammation, and neurotransmitter imbalance, and genetic factors. And previously, we found that metabolism in Alzheimer's disease are very important in the brain. At that time, we found that the microglia, which is the immune cells in the brain, react differently to acute phase and chronic phase. In this metabolism dysfunction is occurred in Alzheimer's disease focused on mitochondrial function, also glycolysis pathway. Interestingly, those metabolism dysfunction is occurred based on those estrogen level. This slide shows that metabolic reprogramming and therapeutic intervention is based on those estrogen signal. Here, this is the premenopausal state and postmenopausal state, and here this metabolism state in normal aging is very smooth. But metabolic state in Alzheimer's disease very rapidly decrease based on those female sex hormone level. So after menopausal, those estrogen level dropped rapidly. At the same time, those metabolic state in Alzheimer's disease brain also showed very rapidly decreased. So also, at the same time, oxidative stress is increased. So all together, those metabolism reprogram is very important for Alzheimer's disease pathogenesis. In that point, those estrogen level is very important to modulate Alzheimer's disease pathogenesis. The next speaker talk about more about those glucose metabolism in sex difference. So Professor Jung-Chan Park is talk about this glucose metabolism in Alzheimer's disease patient system. So here is briefly, in Alzheimer's disease patient has more glucose dysfunction in female subject rather than male, and there are many glucose-mediated pathways are involved in those Alzheimer's disease pathogenesis. So more of explanation is will be done by Professor Jung-Chan Park. Apolipoprotein E, E4, is a major genetic factor, risk factor for Alzheimer's disease. There are three types of ApoE, E2, E3, E4. But those E4 is the major genetic risk factor. And here, if they have two allele of E4, then the age of onset is much, much earlier than none of E4 carrier. Here is a non-E4 carrier. Is 50% is about an age of 90, but if they have two E4 allele, then their age of onset is about 70. So E4 is very bad genetic risk factor for Alzheimer's disease pathogenesis. The role of E4 is neuroinflammation, Abera aggregation, tau aggregation, mitochondrial function. Also, synaptic dysfunctions are related to ApoE, E4. Here is the some epidemiological data to show that if they have ApoE4 in women, then the time course of cognitive dysfunction is very rapidly increased. This is a ApoE4 woman carrier. This is a ApoE4 man carrier. This is ApoE4 non-carrier and ApoE4 non-carrier male. So in this slide, strictly show that ApoE4 carrier with woman has much more rapid Alzheimer's disease progression. So one factor, ApoE4, the other factor is woman. So both together, that makes a severe symptom and clinical implication for AD progression. This is the female and ApoE4 if they have female without ApoE4, and also they have estrogen level, is very higher level of estrogen. After menopause, then those estrogen level is lowered. In this case, they as age goes up, then AD symptom progressed just smoothly. But in this case, ApoE4 with in female, also they don't have estrogen level higher, then those Alzheimer's disease risk increase much higher here. So you can see how those ApoE4 and female sex is very weak for Alzheimer's disease attacks. So as the estrogen role on brain health as a woman age here, sufficient level of estrogen, then that they have many benefits for brain function. For example, synaptic plasticity, energy metabolism, optical cognitive function, neuroprotection. But estrogen level is very low here. Then they trigger hot flash, night sweat, and memory loss, vulnerable to cognitive decline. So estrogen is very important for this cognitive function, especially in women. So here they have a very good comparison schematic graphic. Female left side, male right side, and morphology. First they see morphology, and then they see the different role of those female and male. Here, female had before menopause, estrogen level become highly variable, and this fluctuation initiate estrogen-related dysfunction in metabolic, inflammatory, and sensory processing related molecular pathway. So energy production and age-related gene profiling different, and immune activation, phagocytosis. But in men, they do not appear undergo a peri-menopause equivalent, and in male, the slower transition may mediate age-related dysfunction in male hormonal pathway compared with those of females. So male is very protective for memory function compared to female. So our lab previously showed that gut-brain axis using animal model. Since brain and gut are physically separated, they cannot communicate very well. But if we change the microbiota in the gut, and the brain function become better compared to AD animal. In this study showed that we use the male mice because female mice doesn't work if we treat antibiotics to the animal. These antibiotics didn't work to female animals, so we have to use male. So if we change the gut microbiota, male show the higher responsive than female. So in this result show that gut microbiome is also affected by those gender difference. Recently, 3 Alzheimer's disease drugs are approved by FDA, and all 3 of them are focused on beta amyloid, even though they have different epitope of A beta. And only this one, lecanemab, Leqembi, is approved by KFDA so far. So this lecanemab will be treated to Korean patient. But unfortunately, this year, May, Nature presented that lecanemab is only good for male, not female. So that is very depressive news for female. Also, this AGI, because 2/3 of Alzheimer's disease patients are female, but they said this lecanemab effect is only for males, so they cannot sell those lecanemab to the female patient. So also we think we cannot get any benefit from these lecanemab drugs. So we are very sorry to hear about it. And there are many different kind of insight for study sex difference in Alzheimer's disease, and multi-omics study, including proteomics, transcriptomics, genomics, epigenomics, and metabolomics. So all together, they can figure out the more decisive role of sex difference in Alzheimer's disease. But future direction of sex difference study of AD is to be for precision medicine. To understand the complex mechanism related to sex difference based on large-scale clinical study, collect and analyze various forms of data according to sex difference. Also develop or utilize disease models that can integrate the various forms of data. So these big data can enable the achievement of accurate and diagnosis, treatment, and prevention strategy. As a conclusion, research on sex difference remains insufficient as clinical trial predominantly recruit male participants, while frequently excluding female due to factors such as pregnancy, hormone levels, and the use of birth control pills. Understanding the mechanism underlying sex differences in AD will lead to more accurate diagnosis and the development of effective treatment. Thank you for attention.